Wonderful. With that, it is my pleasure to introduce our first presentation, Single Nucleus Dissection of 9 Alzheimer's Disease Comorbidities, presented by Tristan Sun.
Thank you so much for the introduction. 1 in every 3 Americans above the age of 65 will die with Alzheimer's disease. Despite killing more individuals each year in America than breast cancer and prostate cancer combined, Alzheimer's disease is far less understood than either of those two conditions, and there is no effective way to prevent or treat AD. One factor which complicates the treatment of AD is its heterogeneity. AD predominantly affects older individuals, many of whom have other comorbidities or diseases which occur alongside AD. Each comorbidity alters the progression of AD, meaning that no two individuals have the same AD. Current treatments for AD are largely ineffective because they completely ignore this intrinsic heterogeneity, treating it as a single disorder. By characterizing how comorbidities contribute to the diversity of AD, our work is the first step towards personalized ways to prevent and treat AD. To study how comorbidities affect the brain, we use the ROS-map dataset, which provides clinical and gene expression data for hundreds of donors. One major obstacle we faced was that in this dataset, the presence of each comorbidity is recorded as a single yes or no variable. But in reality, different donors have varying levels of comorbidity severity. To address this, we used the cell-projected phenotypes, or CPP method, developed by our collaborator, Riley Mangan. CPP uses information from all donors to determine what gene expression pattern is associated with a comorbidity. Each cell is scored based on how strongly its gene expression matches that pattern. And we collect the scores from all the cells of each donor and aggregate those scores together to get a donor-level score. The donors are stratified based on these scores into 3 groups. Overall, CPP provides a finer-grained representation of the variation of the severity of comorbidities in each donor. So now let's jump into the results. Because there are many distinct types of cells in the brain, we first asked, which cell types does each comorbidity affect? But before that, let's briefly review the functions of the major cell types in the brain. The excitatory and the inhibitory neurons transmit signals. The astrocytes nourish the neurons and clear waste. The oligodendrocytes and their precursors, OPCs, insulate the neurons. And there are also immune and vasculature cells in the brain. This heat map shows the effect of comorbidities across cell types. Within each row, a darker color indicates a stronger effect. We found that across all the comorbidities, the neuronal cell types were the most strongly affected. However, among the non-neuronal cell types, we also observed that each comorbidity induces its own distinct pattern. For example, hypertension and smoking were found to predominantly affect the astrocytes and the vasculature cells, which is consistent with their known effects on the blood-brain barrier. Meanwhile, head injury, diabetes, and Parkinson's disease predominantly affected the oligodendrocytes and the OPCs. Overall, our results show that comorbidities affect brain cells in cell-type specific ways. So now we've characterized for each comorbidity which cell types it affects. However, we don't know how it affects those cell types. This led us to the question, which molecular pathways does each comorbidity dysregulate? So here, each point represents how a single cell type responds to a comorbidity. For example, these 4 blue dots represent how excitatory neurons respond to 4 different conditions. The stars represent how the 7 cell types respond to Alzheimer's disease. We found that the responses separate into 2 major classes. This led us to the natural question, what drives this separation? And we observed that the separation was on the basis of comorbidity. So those 4 comorbidities on the bottom drive responses which we found landed in class B, while the other comorbidities drove responses which we found landed in class A. And to analyze what drives the separation, we looked at molecular pathways. Pathways are sets of genes with a shared function. Here, red indicates activation of a pathway, whereas blue indicates inactivation. We found that across these comorbidities, which are known to increase AD risk, some comorbidities, specifically the class A ones, induced responses at the molecular level which mimicked Alzheimer's disease. However, some other comorbidities, specifically those in class B, induced responses which were actually opposite at the molecular level. To interpret this pattern, it helps to know how cells respond to stress. So previous work has shown that these comorbidities damage the mitochondria. When cells face this kind of stress, one way that they can respond is by activating mitochondrial and translational pathways to generate new mitochondria. This is what we hypothesize is happening with the class B comorbidities. However, this stress response requires energy. And if there's an insufficient amount of energy, the cells may be forced to enter a dormant state. We hypothesize that the class A comorbidities directly reduce the amount of energy available to cells, perhaps through mechanisms like hypoperfusion, causing and forcing the cells to inactivate key molecular pathways. Future work will test our hypothesis by analyzing different metabolic markers. Overall, we found that comorbidities affect the brain in two distinct ways, which is a previously unknown result. So now, for each comorbidity, we've characterized which cell types and which molecular pathways it affects. But how do those changes affect AD? Previous work has shown that comorbidities are linked with an increased AD risk. On this slide, the number of shaded circles represents how much prior work supports the association. However, these prior studies only show that there is a link, not how or why that link exists. This led us to ask the question, do comorbidities affect healthy brains in a way that causes their molecular response, causes them to become more AD-like at a molecular level? And to answer this question, we looked at how each comorbidity affects healthy brains, and we compared those effects to the changes between AD versus non-AD brains. We calculated the correlation between their fold change vectors over all the genes. And this heat map shows the results in excitatory neurons. Red indicates a very similar response, while blue indicates an opposite response. Across many cell types, we found that thyroid disease induced the most AD-like response. Thyroid disease is a common and modifiable condition which causes hormonal imbalance. So to summarize, we found that comorbidities affect brain cells in a cell type specific manner, that responses separated by comorbidity into two distinct classes, and that thyroid disease is an under-recognized comorbidity associated with AD-like gene expression changes. Little is known about how thyroid disease contributes to neurodegeneration. So our finding about thyroid disease and its similarity to AD was interesting and novel. So where can we go next? We plan to further investigate why the two classes of comorbidities induce these different types of responses at a molecular level. We also want to study combinations of comorbidities. One of the limitations of our study was that we only looked at one comorbidity at a time. However, in 86% of older adults with AD, these patients have at least two other chronic health conditions. So we want to study combinations of comorbidities to more accurately model the real world. We also want to identify the specific thyroid condition associated with the AD-like response. Finally, we want to use the results of our study to identify lifestyle interventions and potential drugs that could be used for personalized AD prevention. I'll answer any questions you might have, and I'd like to thank my mentors, Shanshan Zhang and Manolis Kellis, my tutor, my last week TAs, our sponsors, and RSI, CEE, and MIT. Thank you so much.
Thank you to our speaker. We will now have 5 minutes for Q&A.
It's working. This is a fantastic talk. I was struck by the real separable classes that you have here. So I was curious about the way that one might think about the other aspects of AD that are sometimes described. Plaque formation, tau protein. I don't know if you came across these in your readings, and whether there are other separable sort of phenotypes of AD that fit into these two different classes that you've described.
Yeah. So the question was, we've identified two major classes, but within those classes, are there finer grained separation, and how does this connect to key hallmarks of AD, such as amyloid beta plaques and tau protein? So I wanted to point out that, for example, head injury was very interesting to us because it's known that head injury primarily affects the brain through having a higher tau burden, whereas the other ones are more through the amyloid beta plaques. We also found that alcohol use, which actually is that cluster in the middle you see there, alcohol use was kind of distinct from the other types of comorbidities. So I think it would be very interesting in future work to see how alcohol affects the brain as well, because it's not quite the same as the class A or the class B comorbidities, not quite the same as the other ones.
Well, while you're on this, can you explain this plot to me, how to read it and what the axes are and how you kind of got the result?
Yeah. So the question was, what is this plot showing and what do the axes represent? So for each comorbidity, we looked at how it affects the gene expression of people with healthy brains. And for each gene, we had a fold change. We compared the people with this comorbidity versus the people without that comorbidity. And we looked at how does the expression level of each gene change. And then there are like thousands of genes expressed in each cell. So we had many numbers, one corresponding to each gene. And then we wanted to represent this high-dimensional information in a lower-dimensional space. So we used the TSNE dimensionality reduction method, which attempts to take these vectors in high-dimensional space, but then put them on a 2D plot in such a way that the distances are preserved at a neighborhood level. So that's what this plot is showing. The points which are close together on this plot induce similar responses in terms of the gene expression.
Thank you. So, can you explain to me why you have, on how you actually end up with a class A point that is closer to class B, and why class A is so spread?
Mm-hmm. So one limitation of this embedding method is because we take very high dimensional information and we compress it into a lower dimensional representation. So TSNE actually only preserves the information for points which are close to each other. This plot should not be interpreted in a way like, just because this one seems like this distance is larger than this distance doesn't necessarily mean that in the actual high dimensional space that is a larger distance. It's a limitation of the method we use to visualize the data. Yeah.
If there are no further questions, then we'll move on to our next presentation. Thanks again to our speaker, if everyone could give a round of applause.